So hello, my name is Thomas Dorner. I'm a rheumatologist at the University Medicine in Berlin, the Department of Clinical Immunology and Rheumatology, and I'm also heading a research group at the German Research Center of Rheumatology. I'm very happy to give you some insights entitled, "Is lupus a B-cell disorder?" Very welcome to this podcast, and I will review some aspects, mainly some pros and cons, but at the end, you may forgive me, I will escape a little bit from a clear yes or no. It's really a luxury that we are living in a time where we see by bidirectional translational research how it is evolving, that we have the heterogeneous disease of systemic lupus evolving in one or the other direction. If we look back a little bit in the biography of lupus, we have also seen that patients who contracted HIV at the end of the last 2 decades, most of the lupus and other autoimmune diseases really improved. That's just an indication that most likely lupus is not only a B-cell disease, but also T cells are involved. When we are talking about B cells in general, we need to also take into consideration the heterogeneity of lupus on the clinical, but also on the molecular level. In addition to what we have learned in terms of the classification and diagnosing lupus, that we are using anti-nuclear antibodies as a kind of indicator or signature of the B lineage cells being involved, we also need to take into consideration some of the therapeutic efforts. Initially, by autologous stem cell therapy, but subsequently also with anti-CD52, depleting B and T cells. This and seeing the responses of patients who are otherwise have been refractory, we have already some early indication that lymphocytes, at that time B and T cells, are co-involved in the pathogenesis, because most of these patients really improved, albeit they had some severe side effects, mainly on the infectious side and on the tolerability. Later on, with the introduction of rituximab, at that time by David Eisenberg and the colleagues at UCL, when they combined rituximab with cyclophosphamide, we could see also in otherwise refractory patients improvement. And the latest addition, in addition to our experiences with stem cell therapy, was the early, let's say, reports in patients with severe refractory lupus who received CD19 CAR T cells, where we are going a little bit broader into the B cell system, depleting them, and seeing in patients who prior received rituximab did not respond, that they are really improving, giving some proof that in some of those patients that have been selected, the B cells are really playing an important part independent from the T cells. But if we come back to the question, is lupus a B-cell disorder, we also need to be really clear that we have, in addition to the B lineage signature, also a type 1 interferon signature. And just recently, we proposed a kind of positive feed-forward model with highlighting the interaction of the type 1 interferon signature and B-cell abnormalities. And I think in this positive feed-forward loop, there is an interconnection of abnormally activated PDCs under the influence of immune complexes built by anti-RNP antibodies. Those are firing type 1 interferon. The type 1 interferon, on the other hand, enhances the production of B-cell activating factor BAF, so a type TNF family member, which is mainly produced by myeloid cells, but this increases the survival and abnormal B-cell activation, leading together with interferon, type 1 interferon and BAF, to the differentiation of plasma cells, and those plasma cells are firing tremendous amounts of antibodies, including autoantibodies. To be very clear, we have a kind of positive feed-forward loop of different immune cells with different humoral factors like the cytokines BAF and type 1 interferon, together with T cell cytokines. And coming now back to the aspects of T cells, and before I go into this, I simply wanted to remind you that just the recent obinutuzumab experiences in lupus nephritis already indicated that we have only about 46.4% of the patients who received obinutuzumab in addition to standard of care. And we have more than 50% of the patients with lupus nephritis who did not respond to obinutuzumab as a second generation CD20 molecule. The difference to standard of care was about 13.4%. So standard of care responders were 33.1%. This is in the range that has also been observed with voclosporin, that has also been observed with belimumab in patients with lupus nephritis. This indicates that apparently other mechanisms are in play, and I do not think that only the lack of deep depletion and substantial broad depletion by obinutuzumab is the only reason. We have additional players, and one of the very important players are the T cells, in particular T peripheral helper cells and T follicular helper cells, that on the other hand are able to generate some TB aggregates in the affected tissues, but also activating B cells at the extrafollicular sites. And at the end, it looks like that everything is a common denominator of abnormal B cell activation. But this is related to a lot of other abnormal immune activation processes in systemic lupus, which at the end converge to an abnormal B cell activation. And taking into account the abnormal site of B cell activation in the affected tissues at the extrafollicular sites, together with the hyperresponsiveness of the B cell receptor, and the crossroads of innate and adaptive immunity in B cells, we also need to bear in mind that in particular toll-like receptor 7 and 8 activators, as well as bystander T cell activation, is a very important feature that can lead to abnormal B cell activation. And I think it is time to identify in the heterogeneity of systemic lupus all the different pathways that can converge in abnormal B cell activation, which is a kind of ultimate pathogenic pacemaker, but it's pretty much important for the upcoming years to identify these abnormal pathways of entertaining B cell abnormalities. So, like Shakespeare with the novel To Be or Not to Be was saying, which is a very long story, in lupus autoimmunity, it is very important at the end to re-establish immune homeostasis that can be achieved either by depleting strategies targeting CD20, BCMA, or CD19, but it's also important to delineate the heterogeneity of systemic lupus, where we have maybe not only one, where we have heterogeneous different pathways in the individual patients, and applying precision medicine in order to identify the different abnormalities of the immune system converging into abnormal B cell activation. And at the end, maybe there is not a need to go for depleting strategies, but using other more, let's say, targeted approaches, like we have identified with the inborn errors of immunity, in order to tackle abnormalities of enhanced BAFF, of enhanced type 1 interferon, etc., or using also other principles like that is used in oncology, by tackling the appropriate checkpoint molecules in order to re-establish immune homeostasis without periods of substantial immunoablation. And at the end, I think there is no clear yes and no, but the remaining question or the ongoing question is lupus a B cell disorder, a T cell disorder, or any other immune cell disorder? Will fuel further research, and we are in a great time where we can learn much more from our patients and from the immune system than any generation before us. Thank you so much for listening, and I'm happy answer any questions you might have with the email that is maybe available via this podcast. Thank you so much.